Another really interesting series of studies, more on the simulation side, have focused on trying to understand how the arrangement of hydrophilic groups on hydrophobic surfaces impacts surface hydrophobicity. And so this particular paper out of Reed Van Lane's group in Wisconsin showed that if we take hydrophilic groups and we place them on a hydrophobic backdrop, if we make them completely kind of spread out across the hydrophobic surface, we get a more hydrophilic surface. And if we instead kind of separate a hydrophilic patch from a hydrophobic patch, we actually get a more overall hydrophobic surface. And so in this way, the actual arrangement of the hydrophilic and hydrophobic groups actually make a really big difference. And so from these previous simulations and experiments, we have this expectation that the exact type of charge group chemistry, as well as their spacing and arrangement on the surface, maybe plays this important role in governing the overall surface's hydrophobicity. And so what we really wanted to answer using molecular simulations is, well, how does charge group chemistry and arrangement impact the hydrophobicity of charged hydrophobic surfaces? And also why, right? What is the molecular mechanism underlying these effects? Okay, so to answer this question, we need to start by agreeing on what hydrophobicity even is in the first place. And so today I'm going to define hydrophobicity as the tendency of nonpolar molecules to be effectively attracted to one another, really because they're trying to avoid interacting with water, right? They're trying to avoid disrupting water's hydrogen bonded network. And so from our perspective, we'll think about two different ways of measuring hydrophobicity. The first is what I'm calling the applied molecular view, which is where if we have a surface, we're going to measure the free energy of some hydrophobic solute interacting with that surface. And so the idea being that if it's a strong interaction, this energy well is really deep, then this is going to be a really hydrophobic surface. Whereas if this is a weak interaction, then it's a hydrophilic surface. So pretty straightforward. But we can also come up with maybe a slightly more rigorous statistical thermodynamics view. And this is where, if we look at this binding process associated with going from the solute in solution to the solute adsorbed to the surface, we can decompose this into two separate processes. First, forming a cavity, either in solution or at this interface, and then a second process where we're maybe introducing or inserting the solute into this cavity, or in other words, turning on direct interactions. And so in this way, we can think about the true kind of effect of hydrophobicity as really being this first process, right? What is the delta delta G associated with forming this cavity in solution relative to forming this cavity at this interface? And so I want to think about comparing and contrasting these two different kinds of views, which sometimes agree and sometimes don't agree. Um, so the setups that I'm going to use to probe both chemistry and arrangement are going to look something like this. So we've created a self-assembled monolayer or a SAM surface, and it's going to be roughly about 6.5 nanometers or so. And it's placed as though it was functionalized onto a gold nanoparticle, but in this case, to simplify our system and to make it so that it's computationally tractable to simulate, we've basically removed the gold and we're only simulating the SAM surface. And then we're placing a large column of water on top in order to make sure that the SAMs don't interact with their periodic images. And so we'll look at a series of different spacings of hydrophilic groups on hydrophobic backdrops. So here you can think about these gray dots as corresponding to hydrophobic or methyl SAM strands. And then the blue dots correspond to hydrophilic or charged groups. And so we'll look at four different spacings, S0, S1, S2, and S3, where we're looking at these increasing spacings between these different groups. We'll also have kind of two control surfaces, one which is just a fully methyl terminated SAM surface. And so this will be our kind of idealized hydrophobic surface, and a second surface that is our ideally hydrophilic surface, which is all terminated in hydroxyl groups. And then we're going to study three different types of hydrophilic or charged groups. First, hydroxyl groups positioned at these different spacings, and then amines or ammonium, I should have said, so charged amines, and then guanidinium groups. So the same as we saw in that experimental study. So the first question we want to ask is, what is the free energy of dewetting these surfaces? And to measure this, we'll use a technique called indirect umbrella sampling, where we're literally performing enhanced sampling, but where the order parameter is the number of water molecules at the interface. And so basically by introducing this potential, we're removing water molecules from the surface. And by doing this, we can then back out the free energy of dewetting the surface. So these are the types of results we get out from this. So starting with just the methyl and hydroxyl surface, you can see that all of our free energies are going to be positive. And this is because obviously at equilibrium, these surfaces want to have water near them, right? So it takes a certain amount of energy just to pull water off of them. But because the methyl surface is quite a bit more hydrophobic than the hydroxyl surface, you can see that the free energy barrier associated with removing water from the methyl surface is a lot lower than the free energy barrier associated with removing water from the hydroxyl surfaces. Then if we go to this S0 configuration, what you can see is that for the hydroxyl surface, the surface gets just slightly more hydrophilic when we introduce these three hydroxyl groups. But when we introduce ammonium and guanidinium, it gets a lot more hydrophilic. And in particular, it's most hydrophilic for ammonium, even compared to guanidinium. And then similarly, if we space these groups out, you can see that now all of these systems have become even more hydrophilic. And again, ammonium is more hydrophilic than the guanidinium surface. And so the kind of take homes here are really that increased spacing between hydrophilic and charged groups leads to increased hydrophilicity. So we saw overall things got more hydrophilic going from S0 to S1. And this is consistent with those previous simulation results that showed that if we just spread out all of our hydrophilic groups, we're getting more hydrophilic surfaces. But we also saw that mixed methyl ammonium surfaces are more hydrophilic than methyl guanidinium surfaces. So this was actually the opposite order from what we saw with previous experiments, which showed that the guanidinium surfaces were more hydrophilic. And so to try to understand a little bit more about what was going on here, we decided to look at what water was doing near the surface. So here I'm plotting water density at the interface at a fairly high INDUS potential. So we've removed most of the water from the INDUS cavity. And black refers to high water density, and white tells you where we've removed water from, and the red shows the hydrophilic groups. And so what you can see is that in the hydroxyl surface, we've removed most of the water from the hydrophilic groups. With the ammonium surfaces, you can see that we actually still have a fair bit of water coordinating the ammonium groups. We've removed the water around it, but the ammonium groups are still hanging on for dear life to those water molecules. And then the guanidinium groups are somewhere in between. They've lost some of their water molecules, but are hanging on still to a few. And so this kind of illustrates that while guanidinium is larger, and it actually has more hydrogen bond donors than ammonium, right? If we look at the chemical structure of these two groups, ammonium's stronger hydrogen bonds are energetically much more unfavorable to break, and this looks like it's leading to a higher energy barrier associated with dewetting this surface. So we see this discrepancy between our simulations and the previous experimental results, and so we wanted to try to come up with a simulation that was maybe going to more closely match the experimental system. And so here, what we did was we created a gold nanoparticle and functionalized it with a methyl sand surface. And so this is really similar to that kind of AFM tip in the paper. And then we perform spatial umbrella sampling, and so we'll get this potential of mean force as a function of distance, from which we can get an overall binding free energy. And so if we look at the binding free energy of this nanoparticle to the surface, what you can first see is that we have overall, all of our results are now negative as opposed to positive. So again, this illustrates that this means that we can observe the positive free energy or the favorable free energy of this nanoparticle absorbing to the surface. We can also see that the hydroxyl groups are only slightly more hydrophilic than the methyl group. But again, we see that the ammonium and guanidinium surfaces are way more hydrophilic. And so here I've shown as using an asterisk the PMFs that actually didn't have any minimum. But interestingly, we also see that for the ammonium S0 surface, we actually do get a slightly favorable binding. And this is about minus 5 kcals per mole. So this is not nothing. This is a meaningful minimum. And so this kind of illustrates already that actually maybe ammonium isn't that hydrophilic. We can then also look at the S2 and S3 configurations. And we actually see that as we go from S1 to S2 to S3, our surfaces get more hydrophobic. Our nanoparticle is binding more strongly. And then in the S2 configuration, we actually see again that ammonium is binding to the nanoparticle more strongly than the guanidinium surface. And so overall, we're observing that this S1 configuration seems to actually maximize hydrophilicity. And if we keep spacing our groups out further, we actually observe more hydrophobic surfaces. And then also that our mixed methyl guanidinium surfaces are more hydrophilic than our mixed methyl ammonium surfaces, at least at the S0, S1, and S2 configurations. And so now this looks a lot more consistent with those previous experiments that were published. So again, we wanted to look at what water was doing near this interface to try to understand why we're seeing this discrepancy between what INDUS is giving us and the umbrella sampling. And so here, the main features that I want to point out is that the nanoparticle is binding directly over the hydroxyl groups. And so you can see that the region of white, which is kind of the footprint of the nanoparticle, is encompassing the hydrophilic groups. But then if we look over at ammonium and guanidinium, for the most part, you can see that the nanoparticle is effectively trying to avoid the charged groups, right? It's just kind of working its way around it as opposed to trying to actually de-wet those water molecules. And so even though these water molecules in the solvation shell of ammonium might be hard to remove, they don't actually get removed during the adsorption process. And so this left us thinking, well, maybe the reason why guanidinium is appearing to be a bit more hydrophilic compared to ammonium is actually just because it's bigger. And so maybe the footprint of the nanoparticle on the ammonium surfaces is just a little bit bigger because it can just reach the surface a little bit better. And so to measure this, we actually measured the de-wetted area associated with the nanoparticle on the surface and looked at the correlation with the binding free energy. And you can see that we actually get a really strong correlation with the binding free energy, right? Almost 0.95. And so this indicates to us, we think that that really what's going on here is that guanidinium is just taking up more space on the surface. So this is really interesting, but a lot of the applications we care about are actually related to slightly more complicated solutes, right? And in particular, we care a lot about protein adsorption to surfaces for things like biofouling, protein separations, drug delivery. And so we wanted to think about, well, could we come up with a kind of ideally hydrophobic protein and see if we get the same trends or something slightly different? And so our ideally hydrophobic protein is this protein called hydrophobin, which basically has a completely hydrophobic face covered in hydrophobic residues, and it's also neutral. It does have some polar groups at that interface, but it's mostly hydrophobic residues. And so overall, what we observe is that hydrophobin binds more strongly to a number of these surfaces, the S0 and S1 configurations, than the nanoparticle did. And so you can see that we don't have any of these PMFs that have no minimum. All of them have a minimum. And then the other feature that I want to point out to you is that, again, ammonium is binding more, the ammonium surface is binding more strongly to hydrophobin than the guanidinium surface at S0, S1. And then as we look at S2 and S3, you can see this order reversal. And then it becomes very strong binding at the end. The other thing I want to point out that I think is really interesting is the fact that these hydroxyl surfaces and then some of these ammonium and guanidinium surfaces actually bind hydrophobin much more strongly than just the methyl surface alone or the hydroxyl surface alone. So there's something about having a mixture of methyls and hydroxyls that actually binds hydrophobin much more strongly than just methyl or hydroxyl alone. And so this is just kind of reiterating what I just said. Overall, this was kind of this trend of guanidinium being more hydrophilic was kind of consistent with previous experiments. So again, we wanted to look at the water density. And overall, what we saw, the main take home I want to point out here is that while the nanoparticle really preferred to kind of avoid all of our charged and hydrophilic groups, you can see that hydrophobin actually does interact directly with the ammonium and guanidinium groups, even though it doesn't necessarily have charged residues at the interface. These are all polar and hydrophobic side chains. And so this kind of tells us that even just the hydrophilicity associated with a protein backbone or polar residues is enough to make it a lot easier to remove the water molecules around ammonium. And then similarly, if we look at the wetted area of hydrophobin, we can actually see that now this strong correlation between the delta G free energy of binding and the dewetted area still exists, but it's a lot less strongly correlated. And so again, this suggests that this strong polar interaction between the polar residues and the surface is playing a significant role in binding. So overall, some of our takeaways here were, first of all, that the relative hydrophobicity of these different interfaces depends strongly on your point of view, right? So the energetics of protein nanoparticle adsorption were pretty different than those of dewetting, and in particular, the ammonium methyl surface was more hydrophobic for adsorption, but not when we were looking at INDUS. We also saw that there exists this kind of optimum hydrophilicity or hydrophilic group spacing that results in minimized hydrophobicity, somewhere around the S1 to S2 kind of spacing. And then finally, hydrophobic proteins like hydrophobin seem to bind more strongly to mixed hydrophilic hydrophobic surfaces than hydrophilic or hydrophobic surfaces alone. And so I think I'm starting to run low on time, but just to give you a kind of brief sense of what we're interested in doing next, we're interested in applying this kind of framework to thinking about exploring other types of charged groups and kind of exploring chemical space, particularly thinking about negatively charged groups. We're also interested in connecting this to patterns formed by ligand functionalized surfaces. So a lot of these surfaces are almost artificially ordered, right? And so we're interested in thinking about when we observe patterns form from ligand self-assembly on surfaces, well, how do the patterns that we observe in these really ordered systems translate to those kind of maybe more realistic systems? And then similarly, we're interested in thinking about how did these hydrophobicity of these different pattern surfaces impact our thought about how we think about patterns on protein surfaces. And so with that, I'd just like to thank my group. In particular, Jakir, who is right here in the middle, is really the person who did all of this work. And so it's been really fun so far. And then I'll take questions now, but also if anyone has any questions that come up later or, anything they want to follow up on, feel free to shoot me an email. I'm more than happy to talk more about this.